Hello, in this video I will be describing the process of transcription in prokaryotes and its 3 steps. To begin, RNA polymerase starts as a core enzyme and then the sigma binding factor converts it into a hollow enzyme, which is then utilized in transcription. The first step is initiation. Initiation is comprised of the promoter region, which is made up of the -35, -10, and +1 sequence regions. The -35 and -10 sequence regions are sites where parts of RNA polymerase bind, and the +1 region is where transcription actually begins. If, for example, we deleted the -10 box, which coded for an enzyme to metabolize sugar, it would actually force the RNA polymerase to bind to the +1 region, as shown in the picture, which would cause DNA to begin to unwind. The second step in transcription is elongation. It must be remembered that DNA is transcribed in the 5' to 3' direction. Elongation is shown in the image below. As you can see, it is comprised of the coding strand and the template strand. The template strand is where RNA attaches on the bottom, and as RNA is depicted by the red line, in which makes the bottom the template strand, the top the coding strand. The third step of transcription is termination, and it can occur in 2 ways: row-dependent or row-independent. Row-dependent termination utilizes a row protein that binds to RNA and uses helicase to unlink the DNA-RNA chain, which results in termination. This process is better depicted in the visual of row-dependent termination, where you can see that the row protein will slide up the RNA, which is shown by the orange line, and bind to RNA polymerase, depicted by the red circles, and where the row protein then uses helicase to unlink the chain. In row-independent termination, no row protein is necessary, but it does utilize a termination hairpin, which is made up of inverted repeats that have been joined together. Termination will occur when there is a long sequence of adenines, depicted by the A's, at the end of the nucleotide sequence, which causes the hairpin to break away from the chain. Here is a visual depiction of row-independent termination. It shows the sequence of multiple adenines causes the hairpin, circled in black on the RNA strand, to break away. I would like to credit the lecture slides provided by Christopher Howig for our Genetics 311 class from NC State University to complete this project.